just thinking, ever wonder if you could banish baldness forever?
You sure don't want to look like this guy. In order to prevent hair loss in anyone's life, we first need to understand the process of DNA. DNA stands for deoxyribonucleic acid and is the main resource for all living organisms. DNA is found in the nucleus and is double-stranded. Think of it as a spiral staircase. As we take a closer look of our DNA, we can see it's a double helix structure, thanks to Rosalind Franklin. The DNA structure will only pair up with its complementary base pair. The nitrogenous bases with double rings called purines, which are your iodine and guanine, always combined with your nitrogenous pairs with single rings called purines, which are your cytosine and thymine. Iodine and thymine connect together and create 2 hydrogen bonds, while cytosine and guanine connect together, creating 3 hydrogen bonds. Each base is connected to a sugar and phosphate molecule. Think of a jacket and its zipper. A zipper connects 2 sides to a jacket, just like the 2 strands of the double helix structure. The teeth of the zipper acts as its A, G, C's, and T's of our DNA structure, while the jacket itself is our sugar and phosphate. The zipper will not zip up if the DNA is not paired with their complementary pair, guanine with cytosine and thymine with adenine. When the zipper zips up all the way, nucleotides, which are the patterns creating genes, become a unique DNA sequence that is different from everybody else. Since DNA can leave its cell, it makes a special copy of itself that can travel outside its control center. This is called your mRNA, which is what nucleotides build your A, U, G's, and T's, instead of using cytosine like in DNA. In the mRNA, it changes to uracil, which is single-stranded. Once translated, the 20 amino acids are sequenced, creating building blocks, consisted of different folding patterns to make proteins, which is what shows physical traits like eye color and hair color. This process is called translation.
With the help of Rosalind Franklin's research, James Watson and Francis Crick discovered the structure of DNA. Humans are influenced by nature. Our genetics are inherited from our parents, 23 chromosomes from mom and 23 from dad, which after combining and going through mitosis leads to creating you. Genetics are the blueprint of a person from their looks to their personality, though they can be influenced by surrounding environmental factors. The environment can influence certain genetics based on one's upbringing, for example, their education and social and physical experiences. In a way, nature and nurture work together. For example, some diseases and illnesses can be genetic, but they can also be influenced by the environment. People have genes that increase or decrease their chance of acquiring a disease, but just because one has that gene doesn't mean the environment can't still be able to influence if one actually develops it. This is what epigenetics is, the study of how one's environment can affect the way one's genes are expressed. They do not change your DNA sequence, but change the reading of a DNA sequence by deciding which genes are turned on or off.
In October 1990, scientists began to develop the process of producing maps of the entire genetic makeup of a human being, known as the Human Genome Project. The human genome contains approximately 3 billion base pairs and 30,000 genes. A technique called DNA sequencing is used to construct a map of the human genome. It determines the exact order of nucleotides, adenine, cytosine, guanine, and thymine in a segment of DNA. When determining our genetic sequence, we can understand how the body functions and if there are diseased genes present. When scientists understood genetic makeup, the project's goal was to identify and combat multiple genetic disorders by discovering genes which cause disorders like cystic fibrosis or breast cancer. They were able to make accurate diagnoses by developing new drugs and genetic therapies. Now that we have background information about DNA, we can now ask the question we've all been waiting for. Ever wonder if you could banish baldness forever?
Alopecia is the most common disease that involves abnormal hair loss, which is a major psychological challenge that is affecting people worldwide. We can suppress or even prevent alopecia by using gene modification, also known as gene engineering, which is a process that uses laboratory-based technologies to alter the DNA makeup of an organism. This could include changing a single base pair, like your A's and T's or your C and G's, deleting a section of DNA, or adding a whole new section of DNA. The use of technology techniques such as CRISPR-Cas9 enables geneticists and medical researchers to edit parts of a genome by removing, adding, or altering sections of the DNA sequence.
There have been studies showing that this method is possible. According to the study done in mice by the biomedical journal Scientific and Technical Research, CRISPR-Cas9, a promising approach for alopecia. Saying the Cas9 enzyme is injected into the nucleus and guided by a specifically designed gRNA which navigates the genome, locating at the specific DNA sequence. After locating the target gene found on chromosome AP12, the Cas9 recognizes the sequence and starts its activation process, cutting an incision in the DNA strand. There are 2 outcomes once the DNA strand is cut. 1, it can suppress the function of the alopecia gene by deleting it. 2, scientists can also add a new genetic sequence where the incision happened, modifying the original gene. An example of adding a new genetic sequence is the SRD542 gene, which is inspired by the C57BL6 mice gene that can increase the number and thickness of hair follicles.
In conclusion, there have been challenges in applying this process in humans due to having to be precise in the genetic editing as well as the adverse immune responses. However, DNA technology, especially the CRISPR and Cas9, has promising potential to prevent hair loss in individuals that have alopecia disease, but also other high genetic risks that involve hair loss. We believe that this DNA technology can be possible and aid in hair regeneration with further research and clinical trials.